So, good morning. Welcome to quantitative imaging class in QBI in short. And today I'm going to give you an introduction to what the course is about and also a little bit about images. And what we have and a little bit also about where images come from, how to make an imaging coupling to scientific questions, and a little bit about workflows. Morning. And I don't know if you have already found it, but the course webpage is on this link. And there you can get all the lecture material from lecture notes, so the slides I have here, which are actually Jupyter notebooks, and also lecture recordings and exercises. So you have everything on this landing page. And as I said, these slides are done in a Jupyter notebook, so I need to load some modules. Right now, you don't have to pay so much attention to it, but I needed to run this notebook. And about the course, the question is a little bit about the motivation. So first of all, who are you? Maybe you can, even the break, tell a little bit what you are doing, where you come from, etc. So what scientific area you come from, what will be expected, and of course why this class exists at all. And then we do some collection about different applications and how to work with new computational possibilities, and then comes a little bit course outline. First of all, who are we? So that's me. I studied computer system engineering in Sweden, and then follow up with a PhD in signal processing. Then over three corners, I landed up working as beamline scientist, kind of a physicist. So I did a reverse jump compared to other people. Usually people from physics go into software engineering. And then I worked a little bit as algorithm developer in a company. And that's about me. Then we have also a teaching assistant. That's Daniel, who's sitting back there. And he is a PhD student in the X-ray microscopy group at ETH and at a Swiss light source at Paul Scherrer Institute. And he will help you during the exercises, and also during the projects we will share the task of helping you if you want to do a project. I'll come to that later. Then we have a little bit about the background. This class is targeted at a very diverse background, so we can have art history researchers, we can have chemists, biologists, physicists, also some people from informatics have joined the class, and so on, also mechanical engineers. And also with this wide field of people, we can also expect different levels of skills and complexity. So when it comes to experience working with images, you can have, on some side, you can have the toddlers who know they have images, but they don't know how to continue, up to the mountain climber who more or less know at least some parts of the material, but maybe wants to learn a little bit more about it. Also, the background, some people prefer using software where we can click, mark up things. Other people are more going towards doing scripting. And the approach in this course is that we are rather going towards a scripting because it helps making things reproducible. And also the data, what people bring, can have different complexity. So it can be very simple things, but huge masses of it, which sets one challenge. And it can be maybe not so much, but high complexity, and then you have different challenges. So we try to target both these extremes, or actually all 6 extremes. Let's see how it works out, but anyway, Some may think some parts are dull or too obvious, but other things are a little bit more complicated. Others have more complicated things in the reverse areas. So let's see. So the lectures will be essentially that I'm showing different concepts, a little bit of code, not so much. I try to hide all the visualization code because that can be quite long and doesn't really bring you so much during the lecture, but it's very useful to look at how I do my visualizations, because then you know you can also make nicer presentation of your data, because in the end you have to publish your data and present it. So then you need to fill up all the details about that. But during the lecture I try to hide the visualization part because it takes a lot of space on the screen. The exercises will be done in Python mainly, using Jupyter notebooks. The exercises are laid out in a way you need some basic skill, but already during the first lecture I will introduce you to the basic concepts that you would need, and then step by step it will be added on more things. And well, don't hesitate to ask. We are here to help you. And then there are of course also the opportunities to create your own implementations, in particular if you go for the project part. And there you can really say get dirty on your data. So here we have the exercises. Usually there is one exercise set per lecture. They are not mandatory, but if you want to really get something out of the class, it's good to really work with the data and the exercises. The exercises are laid out as Jupyter notebooks. If someone wants to take the pain of doing the same thing in C++, you're welcome to. I can support it, but I don't really recommend it because it will take you a lot of time to do it. Then we have the project, which is, again, optional. It will be in May, so you will start working on it in May, because then you have most of the material already ready, so you know most of the things. There's only one lecture left, I think, in May. And this project can be either a task that I give, so I will try to provide a list with the different tasks, or, which is even better, you can take your own master project, PhD project, and work on an extracted part of that. Probably not solving the whole PhD during this project. That would be a little bit too much. But anyway, giving you one highlight in your analysis where you maybe have got stuck. And the advantage then is that you will get our support, Daniel's and mine, to get new insights, get an idea how to work with it, and hopefully that will help you to get more forward in your own project. So the idea is that we will have a typical line of questions, go through a workflow, and then in the end, last day of the semester, we will have a presentation where you present what you have done to your colleague students, and then everybody can hopefully learn a little bit more, get a more diverse idea about applications that can be out there. One interesting thing about the project is also that if you want, you can use the project as starting point for the exam. The exam, no, not yet. Let's see. The exam is, did I start? Yes, I did start it. The exam is an oral exam, will be about 25 minutes, so every gets a 30-minute slot, but there is some transition time. And if you do the project, you can choose that we start around the project, asking questions and widen the rings, or else you do a traditional exam where I go through the material that we had in the class. So that is your choice what you want to do. So the overview is, oh, I see here now that this one wasn't updated. Okay, the dates are not correct. That's last year's date. I thought I updated it, but it didn't come through. Okay. Anyway, it's last year's date, but you have the same table also on the web page, which I can show, and that is, hey, come on. There it is. So you have, when you come to the web page, you have the weekly lecture plan, where you have the correct dates. And there is also the notebooks that you can download. The slides will be just a slide presentation of the notebooks, online rendered. Then I will also prepare the PDFs, so it will be written out as a long PDF document if you prefer to read it in that manner. The lecture videos, they will be also uploaded here. They will be on YouTube. There is a specific YouTube channel for the videos. And when there is Spiderweb, then it's last year's lecture. So if you want to watch ahead, then you can watch last year's lecture. There will be minor changes, but not so much. I probably say different things. I don't say the exact same words, but the content will be the same. And then when the Spiderweb disappear, then you have the refreshed lecture. Exercises, you can see there's not all of these last lectures have an exercise. And let's see, maybe I managed to add one more exercise to this part in the end. So go back to the notebook. Oops. So that's the one. So this one I need to update. Sorry about that. And then we have some reading material, some papers, also some web links, and good to know material. I'm not going into depth, but it's like secondary literature. But it can be really helpful to read, and also get a further motivation. This one is about how to work with the data and the code, in the sense that you need to be sure that the code is doing exactly what you want it to. And that is relating to a part that is called unit testing, which I will talk about around lecture 7, I think, or 8. And yeah, there are a couple of more things here, some about cloud computing and concurrency. It's more background material. Then there is 3 books. Actually, yeah, I don't know why I got it like this. That's strange. Yeah. There should be one more book, so I will update that. And these books are available, so you can just download them through ETH library. So it's actually, I think most of them. No, the top one is Wiley, and then there is some also from Springer Verlag. So, and yeah. This one is a pretty good introduction book. It's really a brick. I think when you have the printed form, it's like 7-8 centimeters thick. So it's a lot of material. Gives you a good overview of things you can do, but it's more or less the starting point, because it shows that you can do it. But if you really want to get down into doing it, then you have to go to a second reference. But it gives you a very good overview to browse through. So it's the handbook form. And then I just want to make full screen. No. Why did it do this? So the first motivation is you have data, you did an experiment. Hopefully you didn't strain your hand while doing it, but anyway, you do some experiment, and that produces data, sometimes not so much, so maybe a few gigabytes. In some cases, you have several terabytes. And then is the question what to do when you analyze it, because in that form, it's just a lot of images. Of course, you can watch them, but that will take time. And it will also not be very quantitative. So the analysis is what we actually are going for in this lecture. There is also the motivation. We want to understand the data. So the first steps, you have the acquisition. That's a challenge by itself. But anyway, the level of abstraction is just that you have images. Then you may need to do some reduction, reducing size or changing it from observation domain into analysis domain. Could be that you need to do some transforms to change the physical properties, etc. You may also need to do some reconstruction. Then you do some filtering, but still, it's still just images. There is no interpretation built into it. Then comes the segmentation. Then you start getting some structure in the data, because then you have divided it into different regions. And then you do labeling. Labeling means you tell that this region, you identify each region. So you know that you have, say, 1,000 regions, and from them you can start doing something. Then it starts getting even more, increasing level of abstraction. And when you start doing measurements, so each region, you can do some measurements, if it's the gray level or if it's the shape in some sense or position. And that also increases the level of abstraction. And finally, you do maybe classification. You say that all the items that are round are one class and all items that are squared are another class. Very simplified. And then you can do some statistical. And with that, you increase the level of knowledge for each step. And that is the goal, because when we come to the classification and inference, then you really start to have something that is publishable and presentable in useful graphs. Maybe already the measurements, but yeah. So the last 3 steps, they can start getting publishable. And how to proceed? Well, we can do some click and try approach. So you get your images, you do here some fancy stuff, some magic math, and then you do some other complicated stuff, and then some more math stuff, and then in the end you say ice cream is good for the bones. Well, you can probably do that conclusion, but probably not the way. And using this complicated nested workflow is probably also not a good idea. So you need to do a well-described procedure, and you need to understand what you're doing each step. And this analysis should also happen, should be made in a way that it's easy to do it for 1 image, for 10 images, 1,000 images, or whatever. And the idea is your work effort that you're sitting with a computer shouldn't increase with the same factor, because much of it can be automated. So if you have a good recipe, how to analyze, tested it well, so you're sure that it actually does the same thing, then you just have to write some kind of wrapper around it that does the mass production. And then you can just press the button and come back after the weekend, and hopefully you have a nice graph. It's not always it works that way, but you can at least hope. And if it didn't work well, then of course you have to go back and do some iterations in your analysis workflow. But in the end, the idea is that you should not put the same effort for every image, at least. So we have the experiment life cycle. First, we have to design the experiment. And of course, you have your science area where you know what you want. And then you have to design an imaging experiment that answers some questions that you have. And if you do good planning, you can actually gain some reduce the amount of work when you do the analysis. I have seen example of both people who do experiments thinking in the, say, the traditional way that they do their experiments normally. But it doesn't really work for producing masses of data and a lot of images. So they have maybe to rethink. And if you do it in the old way, then you can come into a very awkward analysis workflow. So you need to plan in ahead to make sure that you have a easy analysis, because the analysis in the end will be the thing that takes a lot of time. Then we have the measurements, where you do the actual acquisition. Can be also a challenge. Later comes an example of producing a lot of bone images, and maybe you even have to make modifications on the beamline or installations at the beamline to automate even the measurement. Data management is a not negligible task, in particular if you start producing terabytes of data, because then you need to make sure that you have, first of all, data stored, backed it up, because in case a hard drive crash, they can. You need to have a backup because beam time is expensive. Then setting up databases, indexing the measurements you have done, and then with these databases and organization of the data, you can better do an analysis. And of course, last part, we have the post-processing, where you do a lot of analysis going through the data with quantitative methods. But it's very time-consuming, and now this is really annoying. I did a lot of adjustments on this slide. But anyway, it says minimal improvements. It actually has been a lot of improvements. With the development of data science, a lot of workflows and ways how to handle the data has helped a lot with post-processing. Not to mention all the machine learning approaches that comes with it. So how much time is spent during the experiment lifecycle? So looking over the years, in the beginning we had some experiment, took some time, then experiment design took some time, then you did the measurement itself, took quite a long time, because at that time detector speeds and the source fluxes, etc., was not so good. So experiments that today are done in a minute took 18 hours back in the early 20s. And then comes the data management. At that time, as you didn't produce so much data, the fraction of data management was also less. And of course, we have the post-processing. Then we have the next. Then you can see that the part for the measurement is reducing and reducing, and even here. Nowadays, it can be a very short time to needed for the experiment itself. Let's say you have a day, you produce several terabytes of data. So that puts a lot of effort into the data management and also on the post-processing. And you can see here that it's essentially the processing part is, plus minus a little bit, 50% of the time. So it's really time-consuming part to handle. So we can produce images at very high rates. Detectors are getting faster. The other day I was looking at the detector that produced 24 gigabytes of data. I haven't seen larger detectors. And they're producing images in very high frame rates. And that is really a challenge, first of all, on the instrument side. It's not so much your problem, but on the instrument side, having the pipelines to slush all the data onto the hard drives. And with the data rates we have here, let's say we produce 8 gigabytes per second, and with that, even on a large workstation, you would saturate most of the analysis tools if you want to push everything into the memory. Therefore, you need to have workflows that can handle data coming in, data coming out. So you need to build that into the workflow. Then, Again, there is a link. One of them is not available anymore, and the other one is actually from 2013. So things have happened. One thing is that Instagram produce a lot more data, but also that the cameras produce more data. So things have changed since 2013. Then we have different sources, how the images are produced. So we have, for example, X-rays. You can have the laptop, desktop X-ray system, but you can also go to a synchrotron, and there you produce a lot of data. For example, at the X-ray tomography, you produce 1,000 frames per second, and that would give you 8 gigabytes per second. In CSACs, produce maybe 30 gigabytes per second, and on a nanoscope beamline, 10 terabytes per day. So it's really, really large amount of data, and also the files. A file which is 500 gigabyte, you can't even get it into your computer, so you need to read it in pieces. And then we have optical systems, where you do, on one side, you do just normal microscopy, and you can do light sheets in microscopy, 500 megabytes per second, or confocal images, the 78 megabytes per second. And then you have the personal equipment. So, for example, the GoPro action camera produces also 60 megabytes per second. And that's also pretty inexpensive device. All these, they cost quite a lot. And then there is another 10 frames per second, 1000, which also is pretty cheap. Doesn't do large images, but it produces still a lot of data, 400 megabytes. So even the consumer electronics is capable of producing a lot of data. So handling masses of images. So XKCD, favorite, has a lot of good knowledge in it sometimes. So in principle, when you start analyzing data, it's one thing that you have the data, but as soon as you start processing it, you're creating more and more data. So actually looking at the graph that I had before, or actually the topics I had before, we have the acquisition that produces the base amount, but then with the reduction, you may double it, because you just reshuffle the data in some sense. Then you do reconstruction, still adding a lot of data, filtering, then you want a new copy, and so on. So the data explodes when you start analyzing it, and that's also something to consider. In the end, when we come to the measurements and classification, we are more talking about tables. Then it's easier to handle, because then it may just be a list of some kilobytes, and that's not so bad. But in the beginning, the first steps, you will really produce a lot more data. So, thinking about what is a terabyte. So if we look at this image, it's just a noisy image, is 1000 by 1000 pixels. And say you can inspect 1 per second, which is a pretty good frame rate to inspect it. Still, that would be that doing this calculation, we have 1 terabyte, we have the image size of some pixels, and then we have the hour, and then we do a calculation and come to the point that we have 1 terabyte divided by the image size divided by the number of hours. And then to watch 1 image per second in a terabyte takes us almost 140 hours to go through manually. So we really need to put the computer on doing this. And the computer would, terabytes, that takes a little while still, but at least it's not 140 hours. And looking at the data, so the first one I showed was just noise, but what we want to look at is more structures or some features in the images. This is a slice from a bone, and we want to identify and make some assessment about these images. So we can say, first of all, we want to ignore the large ones, like these and the long lines here. They are presumably just segmentation artifacts. But we want to see all the other ones, and we want to see, maybe categorize, are they more on the left side than on the right side? Or are they larger on one side than the other side, or up, down, whatever? We want to cluster it and say something about sizes. And with 1 second per image, that's quite a big challenge. You probably would need more. The more questions you have, the longer time you would need to make this assessment. So this is a single slide. Now, let's do it a bit more tricky. Now we want to do it over 96 samples, and to make it more fun, we also do it in 3D, where each sample has 2,000 slices. Then it's getting a bit tricky to analyze, in particular also looking at it in short time. The three-dimensional feature makes it harder to make an assessment. You can, of course, with the visualization, do some enhancements, making it easier to analyze, but still, it takes a long time. Then, to make it really tough, then we go to a great population, because a biologist wants to have a great population to see variations between different treatment categories, etc. So now, doing it on over 1,000 samples, first of all, the challenge is how to measure. 1,000 samples going in and out. Let's say one scan takes 10 minutes. Then you have to go in and out 1,000 times, which is quite a long time. And going in and out also costs. So what they did for this experiment, they had a tray of samples and a robot that took each sample, scanned it, took it out, next sample, scanned it, took it out. So they really need to build extra infrastructure in order to make this big analysis or big scanning. Then comes the next question, how to analyze all this. Again, it's a huge amount of data, and we want to, in the end, tell something about the structure in these bones. And just to make it more fun, there can not only be very strict measurements on the images, but there can also be more soft, fuzzy descriptions. So, for example, looking at the alignment, putting more description names on it, so it's kind of aligned, or well aligned, or not really aligned. So that is also possibly words you need to work with, and then you also need to have metrics for putting it into these categories, probably with some probability. And another topic where it's also hard to do the analysis is if you have dynamic information. So if you look at this video, which I see, oops. This is, what you see here is the space between bubbles. So it's more or less the skin of the bubbles, and the question is to investigate is if these bubbles have the same shape, if they change shape when they move up, if they are different shapes in the center compared to the wall, and if they are rearranging or maybe merging. So questions like that also could be assessed with the image analysis approach. So let's bring in some scientific method. So first of all, what is the purpose? You want to discover something new and learn something new, and of course also validate it. So it's starting out with you having a hypothesis, and that hypothesis needs to be validated. So you need to use this approach to convince the scientific community that this is actually a true hypothesis, or failed hypothesis can also be. And of course it needs to be built on the experience of other ones, so we don't have to start from the beginning. One side is the publications that you can read, how people did it. In some cases you can also get even pre-made source code, which is even better starting point. If you get the source code from someone else, of course, you need to validate the source code, making sure that it's doing what you want, and maybe you need to modify it. And of course, we can do qualitative assessments, but in the end, we need to do some quantification, and the quantification is what we put in our graphs. And the idea is to get these numbers, you also have to describe something it in a way that other people can reproduce it afterwards. And science and imaging. So we have seen that images, they're very easy to make a qualitative assessment on, but how can we interpret them quickly and hopefully not too much programming investment? Because programming takes time, you have a lot of debugging, you have a lot of testing to make sure, but it is possible to do it because there's a lot of libraries already written. But just comparing to process some scalars quantitatively is quite easy, but with images, pretty much harder. So, for example, if you take the thermometer and measure the temperature, it's easy to tell we have 50 Kelvin, plus minus something. But if you do measurements on images, it's much easier to do mistakes, errors, even choosing the levels where you set, for example, a threshold, which I will talk about at some point later. It can have an effect on how large you measure things. And also small variations in parameters can have a big effect and produce confusing analysis. And with image processing, people like to do coding, so there are thousands of algorithms available, some commercial, some open source, and also very many PhD students who work on their image analysis project have written some degree of their own tool for doing the analysis, and therefore there are also thousands of tools. Some may be reusable, but many not. And also the analysis of images requires not only a single calculation, but usually a sequence of measurements or calculations to come to the final result. And of course, experimenting is always time consuming, also on the computer. So you have to have a good idea where to work with, how to work with the data. So, nice renderings, which is what you can get from imaging, is of course interesting. It's good for your presentations. It can also be good to have as an eye catcher for your publication. But often that doesn't tell so much. In this particular case, it was actually through visual analysis that we got the answers. So this is little gold piece from the early Viking Age, and the archaeologists, they wanted to know how this loop was made and fixed. And through the visual analysis, we could see that it was just folded and clamped. So there was no soldering, and that was enough. In the cultural heritage, often that kind of analysis is sufficient. But then this is another case, which is also from cultural heritage, which is about corrosion in steel. So what you see here is the gray level distribution for neutrons and X-rays, and with that you can make conclusions about what is corroded, what is steel, and what is just soil. And that was a much more complicated validation of conservation procedure. So there are different, even though it's within the same area of research, you can still have very different types of analysis. So the initial questions are pretty important to specify also in the experiment design. So what I'm looking for can be look like an obvious question, but it's not always that it's so obvious. And because next question here is my original question too specific or too general. And if you ask it in one way, which would maybe be natural to you, it may be too specific to be able to reformulate it into an image analysis question, an image analysis task. So that brings us to the next question, is how to plan the experiment in a way that you can do a good analysis and reliable analysis. And with this class, I hope to give you a little bit of the language to help you to translate your original question into a question that can be analyzed or in the vocabulary of image analysis. And often you can have some a priori information, and that's really, really helpful to bring into the analysis as well. So if you can have things that help, then of course bring it in. But it needs to have some correlation with the data. The classic example is that the health insurance connected to some disease, there is no connection between them. So in that case, it's irrelevant information for the analysis. It may tell how feasible it is to treat the patient with the funding, but it doesn't tell anything about the disease. So what we want to do is, for example, compare different treatments. We want to understand and model the process. Of course, that can come back to that one. We also want to inspect components. So seeing if there are faults in it, like cracks in a cast, for example. And these are the, say, top level tasks that we want to do. But bringing in what the purpose is of the experiment, so you can actually, that was also reshuffled. Sorry, that was already said. So what can we measure with the image analysis? So what we have actually two main categories, and of course the mixed category. One is quantification through the gray levels. And with that, we can see material composition, we can see material transport, or else we can also go for quantification from shapes. So identify and count items. We can measure volumes and of course characterize the shape if it's more round or elongated, etc. And then we can look into finding hidden objects. We can measure dimensions, we can count mixing, track changes, and so on. And that is all the main things we can measure through images. And why quantitative? This is a classic visual illusion, optical illusion example. Looking at these two boxes here, which of the center ones is brighter. And the optical illusion tells us that this one is brighter than that one, but in reality, they have exactly the same intensity. So the visual analysis can really be tricked out by such effects. So for really knowing this, we need to do some measurements and do some statistics, and then we can say, okay, they are the same. Another one is looking at gradients. In some cases, it's easy to tell that this is a stepwise gradient, but the more grade levels you have, the harder it is to really see the difference in grade level. So if this one is grade level something and the other one is grade level something else, it's really hard to see what is the difference. And of course, finding the right tool is always the question. Should I take a hammer or a saw? Who knows? But in principle, you have a couple of criteria to work on. First of all, how many samples and how complex are the samples? Is human interpretation or interaction needed in some sense? What is the end product? What you want to really have out of this data? And are there already tools available? That is also quite an interesting question. And will there be more similar data sets? So will you produce more data sets, or will your colleague produce similar data sets? And that gives you the choice between different alternatives. One is maybe there is already an interactive tool. Otherwise, you may do some scripting using existing toolboxes, and if the task is really tricky, then you may need to have develop your own algorithm. But this cartoon here tells also about the danger. You think you improve everything by writing some nice optimization tool, but in the end, you may land in something ongoing development which you can't really control anymore, and then you spend a lot more time in doing. So I would say, actually, this would be the order of selecting how to approach the problem. Probably with emphasis on the middle one, because then you have the possibility to do repeated tasks easier. And with that, let's just see how many slides do I left. I finished this topic and then we do a break. Oops, sorry, wrong way. So what we want, no, I did right. So, good. So, in scientific experiment, you want some kind of reproducibility, which would be fantastic. So that means someone does an experiment, or you do an experiment, you do some analysis, and describe it in your publication, and then from that, someone else should be able to take from that description, be able to do their own experiment and also their own analysis, and then you should produce the same result within some error margin. Repeatability is less strict because then both work on the same data and also the same analysis, but there are different persons doing it. So then we have more or less the human factor and the choices, and then you should compare afterwards if it's the same. And that's less strict, but it's also easier to handle because it's the same data or possibly different data, but the analysis block should be the same. So you should be able to produce the same results. And what is demanded is at least repeatability, but if you can, reproducibility would be fantastic. And with image analysis, different noise conditions, it is pretty hard to really get this. And well, as soon as you measure things, natural variation, everything can be a pretty big challenge. And also with the multi-step approach on image analysis, there are many parameters to tune everywhere, and could be quite time-consuming to go through everything. And how we can keep track of things is, first of all, it's very important to document the analysis steps, and also the code you write should be well documented and understandable for someone else to take up. If you start writing very nested code, it's really hard to take over and also to maintain for other people. But there's also a topic that we will talk about in a later lecture. And with that, I think we'd make the break.